So we'd gotten through mitosis last time. And mitosis, if you had to tell somebody what mitosis is, my parents are always asking me biology questions. My dad was in the 8th grade for 34 years. He loves to say that to people. He taught American history. So he loves to ask me these questions. So if my dad asked you, what is mitosis? What's something very simple? You could even use one word. Okay, dividing of cells. Okay, that's good. How do the cells divide? Do they divide equally, unequally? Equally. And what do the two cells look like after they divide? Identical. Do you remember the one word I used that could describe that process? Sounds kind of like science fiction a little bit, but cloning. Mitosis is cloning. You start with one cell and you end up with two identical copies. Now, do you remember what cells in your body undergo mitosis? Somatic cells, body cells, most of them. Most of the cells in your body undergo mitosis. It's cloning. There are only really two cells that undergo this process of meiosis, one in each gender. And what are those cells? The sex cells. That's a good way to describe it. Another fancy name is gametes. Gametes. Sex cells is fine. No. Sex cells is fine. I'm fine with that. Because that's also correct terminology. Gametes are sex cells. Either the sperm or the egg. And the difference between cloning and mitosis and meiosis, what do you end up with in meiosis at the very end of the process? Are the daughter cells clones? No. How are they different than the parent cell? A basic difference in the daughter cells after meiosis and the parent cell. Huh? Thought I heard somebody say it. Chromosomes? What about the chromosomes? 23. How many did you start with? So you have cut in half. And do you remember we said meiosis was a reduction division? So we went through the process of meiosis 1, this first division. And what is it that we are segregating into our daughter cells in meiosis 1? I used that word very carefully last week. Segregating. What are we segregating into the daughter cells in meiosis 1? Homologous chromosomes. Now, somebody over here, I usually talk to this side. I don't mean to leave y'all out. Somebody over here on this side. Give me sort of a simplistic understanding of what is homologous chromosomes. What does that mean? Somebody tell me what homologous chromosomes are. The same, homologous, right? That's a great place to start. So these chromosomes are the same. Can you kind of give me an example? Okay, now, here in our division, when we segregated the homologous chromosomes, they already exist as sister chromatids. But remember, in the human genome, we've got 46 chromosomes, but are those 46 completely different from each other? The 46 exist in pairs, right? So homologous chromosomes are the same in the sense they are the pairs of chromosomes. Chromosome 1, chromosome 2. They come in pairs, and those pairs are called homologous chromosomes. That's a good job. Now, we've moved homologous chromosomes that still exist as sister chromatids. They're still stuck together with their duplicated equal. And so here in meiosis 2, we're going to take those now two daughter cells that have those sister chromatids that exist as those homologous chromosomes, and we're going to pull them apart, and it's going to look just like mitosis. Now, once again, remember, between meiosis 1 and meiosis 2, we do not copy our DNA. If we did that, it would screw the whole thing up. We would end up with 46 chromosomes in our daughter cells. But what we're going to do in meiosis II, in each daughter cell, we have 23 sister chromatids, and we're going to pull those apart. And so at the end of meiosis II, we're going to have 4 daughter cells, each containing one set of every chromosome. So there's going to be a total of 23. And you remember that term that we used last week for chromosomes, just that had one N number of chromosomes, one copy of each? We said diploid where you had 2 copies of each. But if you have one N, it's haploid.
So at the end of mitosis and meiosis I, you end up with 2 diploid cells. And at the end of meiosis, you end up with 4 haploid cells.
All right, now if we're just counting number of chromosomes, I would have to say yes. But remember, at the end of meiosis I, we have 23 sister chromatids. They're still attached together. And so one is attached to its identical copy. So in that sense, scientists still look at that as a haploid cell. And maybe because they're still stuck together. I don't know. All right. Because in mitosis, we line up those 46 sister chromatids and pull them apart. Here, in this meiosis II, we're going to be lining up the 23 sister chromatids. So we already have half the number of chromatids. That's a little gray area, so I don't want to make that any more confusing. That is not something I'd be asking you on the test, that particular question. Okay. So here is meiosis II. What are our 4 stages? PMAT. Who said PMAT? Good job. Fist bump you if I could. PMAT. What does P stand for? That's the first one. What is M? Metaphase. A. T. Telophase. That is for mitosis. That is for meiosis I and meiosis II. In meiosis I and II, all you have to do is put PMAT and then a 1 by prophase 1, metaphase 1. Meiosis II, prophase 2. You see the difference? You designate which cycle you're in. So I'm not going to go through all the stages again because the characteristics are the same. Prophase 2, spindle apparatus forms our sister chromatids. They're highly condensed. Metaphase 2, all of the chromatids line up. Again, it's going to look just like mitosis, except we have half the number. Anaphase 2, we pull the sister chromatids apart. Telophase 2, the chromosomes arrive at the poles. Cytokinesis begins, and at the end there are 4 haploid daughter cells. 1 copy of every chromosome. 22 autosomes, body chromosomes, and 1 sex chromosome. So that's our 4 daughter cells. So again, prophase, because we're not at the midline yet. Spindle apparatus, sister chromatids. I mean, this is the same stuff, so you don't have to write all this down. Because again, what does it look like? Same steps, same characteristics as mitosis. Metaphase, lining along the equator. How many sister chromatids are lining up at the equator in meiosis II? That would be a good test question. How many sister chromatids line at the equator of meiosis II metaphase? 23. 23. Anaphase, 23 sister chromatids are pulled apart into their 23 separate chromosomes. The spindle apparatus not only is reeling in the chromosomes, but is pushing to elongate the cells. And once we get to telophase, cytokinesis separates the daughter cells, and we reform the nuclear division, and we have those 4 haploid daughter cells. Meiosis I and II, kind of the same. Yes, the difference is in meiosis I, what are we segregating to our daughter cells? Homologous chromosomes. And those homologous chromosomes occur as sister chromatids. They're still attached to their copy. Meiosis II, what are we now separating into our daughter cells? the sister chromatids and the chromosomes. We're pulling them apart here.
Okay, so those we separated in meiosis one, and now we're separating those.
Correct.
Okay.
Yeah, we moved the sister chromatids into cells in meiosis one. Now we pull the chromatids apart in meiosis two. Okay, got that? And that's really the only difference that we see. Now, we're going to modify what I just said just a little bit as it relates to sperm and egg. Sperm formation, spermatogenesis. At the end of spermatogenesis, which is fancy name for meiosis, we're going to get our 4 haploid germ cells, and these are called spermatids. They don't really mature until they get into the male reproductive tract, just about at the point of ejaculation. And in fact, the sperm in male ejaculate cannot fertilize an egg. For in vitro fertilization, those sperm have to be treated as if they're in the female reproductive tract, because there's certain enzymes, certain proteins that activate the sperm in the female reproductive tract. It's called capacitation. So they have to be treated first. Now, in the female side, the process of forming an egg is called oogenesis. But at the end of this process, notice, you do not produce 4 eggs from every parent cell. At the end of meiosis, during this process of oogenesis, you produce 1 egg and 3 polar bodies. Polar bodies are essentially a nucleus with genetic material in it. These 3 nuclei have 23 chromosomes each. The egg has 23 chromosomes each. So do you see how they're like haploid cells? But with a polar body, you don't really have any cytoplasmic material at all. All the cytoplasmic material stays with the 1 egg, which can also be called an ovum. Because remember what we said at fertilization. What do you get from dad? 23 chromosomes. What do you get from mom? Everything else. 23 chromosomes and all the mitochondria, all the Golgi, all the ribosomes. And one reason all the cytoplasmic material stays with the 1 egg is the first couple of divisions after fertilization. The cells don't have a chance to grow. You know that interphase cycle? Do you remember interphase? And we had the G1 and the G2. What does the G stand for? Growth. So the cells don't have time to grow. So with these first divisions, they're divisions without growth. They're called cleavage divisions. So since nature has set this up, you know that the cells can't grow, so they don't get too small. You got to start out with a bigger one. And that's why you have 1 egg produced during oogenesis, and the 3 other packages of genetic material are just discarded. Because you don't need that stuff anyway. All right? So here again is our spermatocyte. This is going to come from our early gamete-forming cell, the spermatogonia. Come down, boom, there's our active spermatozoa once we put it in the female reproductive tract. Here we have our oocytes. These are going to be sort of examples of what's happening in an ovarian follicle as that matures. But as we undergo the process of meiosis, we're going to end up with our 1 egg, 1 ovum, and there is showing, this line's pointing to, you can't even really see them here, but 3 polar bodies, just those packages of genetic material that are discarded. They're just thrown out like trash. And then we end up here with our large egg, 23 pairs of chromosomes, and our other 23 pair are going to come in. And as soon as fertilization happens, those 23 chromosomes are going to be replicated into sister chromatids, and they're going to line up right along the middle of that cell. And what is that going to look like? If you line 46 sister chromatids up along the midline, what's that? Metaphase of mitosis. So when fertilization happens, it's almost as if it started in the middle of that first mitotic division. And then the cell divides into 2, and those 2 into 4, and da-da-da-da-da. On and on and on. All right. That's mitosis, meiosis. Now, all this genetic material that we've moved around, we have segregated into our sperm and egg, or we've made new cells that are growing and dividing. You remember, especially in mitosis, in interphase, we had the G1 and the G2, and we had this stage in the middle. What's the stage that's in the middle of G1 and G2? Synthesis, or S phase. And what is being synthesized during the S phase of interphase? What's being produced there? Or what's being replicated? What's being replicated in the synthesis stage of interphase? New DNA. It's in the S phase. We go from being a chromosome to being sister chromatids because we made our copy. And the reason we did that is our daughter cells have to have that genetic instruction because we've got to use that to make proteins, right? That's what DNA is all about, making protein. And so now when we look at our DNA, instead of focusing on, okay, we've got the DNA and we're putting it in our daughter cells so that they have that exact copy, now we're going to see how we really put the DNA to work. And so we didn't talk much about DNA replication. There's a lot of steps and a lot of enzymes that are involved in doing that. We just clumped that into S phase. We just had this little box, S phase, and that's where replication occurs. Replication is where you form your sister chromatids. What we're going to focus on now for the rest of this lecture is transcription and translation. Remember, I started kind of trying to get you familiar with those terms. Transcription is where we make a copy of the genetic code that exists as DNA. We make a copy of that in the form of RNA. Now, where does your DNA live inside your cells? It lives in the nucleus. Where are we going to use that genetic code to make proteins? Where's that going to happen? Does that happen in the nucleus? It goes out, and the protein is made in the cytoplasm. That's why we make our RNA, because it's smaller. It can get through those nuclear pores. And it can get out to the cytoplasm where it's going to interact with one of our organelles, one of our non-membrane bound organelles. Do you remember which organelle I said was the protein factory? The rough. And why is it rough? Ribosomes. It's the ribosomes that are the protein factories. The ribosomes can either be free-floating or on the ER. But that RNA is going to interact with our ribosomes, and that RNA code is going to be used to assemble our amino acids into protein. And again, that happens in the cytoplasm. Translation happens in the cytoplasm. Transcription happens in the nucleus. That RNA molecule is like the text message that can come out of the nucleus and go to the cytoplasm. So this is a nice synopsis slide covering both of those processes in getting that DNA code transformed into a protein. Now let's look back just a little bit. Nucleic acids. We have two types of nucleic acids, right? DNA and RNA. DNA has our double-stranded molecule that twists around itself. And it does so in such a way that we have a little tiny groove between the two and then a fairly large groove. This is called the major groove and the minor groove. And it's going to be between some of these that we can actually form and start the formation of our RNA. Start that process of transcription. Now, in this illustration of our double-stranded DNA, you see we've started pulling our parent blue strands, we've separated them. And do you remember what kind of chemical bonds holds those two strands of DNA together? Hydrogen bonds. And why hydrogen? Is it the strongest bond? The weakest bond. You can separate it. You can separate it pretty easily. So we broke those hydrogen bonds between our nucleotides, A, T's, and C's, and G's. And so with our DNA, each strand is a template. When we're wanting to replicate our DNA for synthesis of mitosis, we're going to make two new strands. And so now we've got these two copies. These two copies, each one's going to go into a daughter cell. But notice each daughter cell is going to get an original and a new one. That kind of slows down the error rate if each daughter cell is at least getting one original copy of our DNA. But to be able to produce our RNA molecule, we're going to have to unzip that DNA as well. All right? So, while we're not going to get into the big details of DNA replication itself, I do want you to understand a few things. And breaking those hydrogen bonds, those weak little bonds, it's not going to take heat, it's not going to take some other kind of chemical, but it's an enzyme. Anytime you see ASE on the end of a word, that typically designates an enzyme. And in this case, helicase means this enzyme is breaking the helix, the double helix of our DNA. So helicase is the unzipping enzyme. If you look at your zipper, it's sort of got that double row of teeth. And as the zipper moves up or down, separating those teeth, that's what's happening here with helicase. It's breaking our AT pairs, it's breaking our GC pairs, so that we can then begin the process of replication. Now, as we make our strands of DNA, some are going to be long, some are going to be short, and that process begins all along the length of our chromosome. We're going to have a lot of pieces that we have to glue together. And the gluing together of our DNA is going to be by a molecule called DNA ligase. So helicase unzips. Ligase, to ligate something together means to put it together, glue it together. So DNA ligase joins all of our fragments that have been produced together. But the enzyme that's making the new strand of DNA, that new strand that's going to produce these that are glued together with DNA ligase, it's called DNA polymerase. Because we're basically taking our monomers, and do you remember what our monomers of nucleic acids are? What are our monomers of nucleic acids? This is actually the easy one. Nucleotides. Nucleotides are our monomers. DNA and RNA, those are our polymers. So if you're assembling the monomers into polymers, it is a polymerization, right? So, DNA polymerase brings the nucleotides, starts sticking them together in order based on what it's reading from the parent DNA molecule. So let's go back here. So here in purple we have that parent DNA molecule. And our DNA polymerase, we'll say it's right here. That DNA polymerase says, okay, on the parent we have a G, so that means I have to stick a C on this strand. Here we have a C, I'm going to stick a G on this strand. So the DNA polymerase is basically reading along the parent strand and using that code to assemble an opposite or complementary new strand. So when the DNA polymerase reads an A, what is it going to put in the new strand? A T. Reads a G, what are we going to put? It's going to be a C. When we get to a T, what are we going to put? An A. Just say we're doing, this is DNA right now, we're going to put an A. Yeah, we are going to change that when we get to RNA though, so I'll call on you for that one when we get to it. Now what's cool is DNA polymerase has something called proofreading. And do you know that the college student spends much, much less time proofreading a paper than you do writing it in the first place? Anybody else hate proofing your paper after you write it? We agonize over writing. We put it off until the last minute. We throw it together, and then we want to be done with it. Where you could get the most value for your time is doing just the opposite. Take time to put together an outline. Write the paper fairly quickly, and then go back and spend a lot of time proofreading the paper. That's how you're going to really refine and get your paper. But our DNA polymerase has proofreading. So every time it's reading along, so I'm DNA polymerase and I'm reading the parent template. Here's a G on the parent. What do I need to pull out of my bag and put in the new strand? A C, okay? Here's an A. What do I do? What do I put down for an A? A T. Well, before DNA polymerase moves on, it checks back. Oh, there's a G. I matched it with a C. Okay, I'm good. All right. Here's a G. If I put a T down there, that's a mismatch, right? That's incorrect. So as DNA polymerase would go on, it proofreads back, sees it made a mistake, and like the garbage truck, beep, 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 it backs up, removes the incorrect match, and fixes it. So the error rate for DNA polymerase is something like 1 to the 9 to the 10th factor. 1 in about 10 million nucleotides it's going to put down before it makes a mistake. That's a good thing, right? Because if we get a mistake in our gene in the wrong place, could mean cancer, could mean sickle cell anemia, or some other horrible illness and disease, because it would only take 1. So it's a good thing that our DNA polymerase is pretty careful. Now remember I said each daughter cell gets 1 copy from the parent. And they get 1 new strand that was just made. That's called semi-conservative. And as I mentioned before, the value in that is you know the parent strand is a good strand, right? How do you know the parent strand is a good strand and has a good code? This is kind of a logic question. How can you know?
Can the letters be mixed up?
Well, they can't be mixed up. And how do we know they can't be mixed up? The parent was able to reproduce, right? If this was jacked up, the cell wouldn't be around to reproduce. So since it can replicate, can it go through mitosis, it must be good code. So we want to make sure that good code, at least 1 copy is going into each daughter cell. And use that good code then to build a template from. So again, survival of the species, that's kind of what all this all equates to. And so that semi-conservative replication is extremely important. Now, that was just DNA replication, very quick. Very, very superficial. Normally we can do a whole lecture just on that. But I wanted to introduce that a little bit because transcription, remember producing RNA copy from DNA, is very similar to what we see there. RNA is a polymer. RNA is made up of our 4 nucleotides. And what are our 4 nucleotides of RNA? G, C, A, and U. There you go. I was waiting on you for that one. Uracil. Uracil replaces thymine in RNA. You will never find uracil in DNA, and you should never find thymine in RNA. Okay? Now that's how it's the same. But transcription is different in the sense we can't use DNA polymerase because we're making RNA. In that case, we use RNA polymerase. And our copies of DNA that we make, each copy is going to be double-stranded. 1 original strand and 1 new strand. But for our RNA, we're going to be making a single-stranded copy. So it's a little more simplified than what we see with DNA replication. And in fact, we use several enzymes. We used helicase for DNA. We use ligase for DNA. Here, RNA polymerase is kind of a one-stop shop. It pretty much does just about everything itself. So in this illustration, you see our double-stranded DNA. This sort of dark bubble that you see, it says assembly site. You can write that in, RNA polymerase. RNA polymerase is going to attach, begin to unzip the DNA. It's going to help hold it open as it assembles our nucleotides into this single strand of RNA. And once we finish making that copy of RNA, it's simply going to float away. The RNA polymerase pops off and the DNA just twists itself back together. And so here we can see our double-stranded parent, which we can think of as the template, and here we see our RNA copy. And we're going to change some things about this a little bit, but you understand what we've just done. Instead of making a new copy of DNA, we've got our copy of RNA, and there are U's. There's a U, there's a U. So we know this is RNA. If that was DNA, it would be double-stranded, we'd see T's. Transcription, RNA copy from the DNA gene using RNA polymerase. Right? So we've made this copy of RNA. Now, on the previous slide, some suggest it's a primary transcript. Some say it is an immature transcript. It's not finished yet because it's got some pieces to it that we not necessarily going to use. And so what's going to happen is a process called splicing. Now, you and your generation, you don't have the joy of having a favorite cassette tape. Okay, I'm not old enough to have 8-track tapes, but a cassette tape that you listen to over and over and over. You don't have the joy of that thing breaking or mangling up and getting eaten in your player, in which case you have to cut out pieces that are messed up and using Scotch tape the ends together, splice them together. Well, that's what we're doing here with our RNA molecule. We're going to have two components. We're going to have the good stuff. The good stuff is called exons. These are going to be the genes and the code we're going to use to build a protein. But nature has designed our genes to also contain sequences that don't do anything. They're in between the good stuff. And the in between stuff are called introns. So before we can put the full code back together and use it to make a protein, we got to get rid of the junk. And the junk being introns mean we have to cut out the junk and glue the exons together into our mature final copy of the code that we're going to use to make our RNA. Now, let me see. We'll go back to this picture just for a second. You see how this says primary transcript? And you see the blue, the dark blue are the exons, and this lighter kind of pink are the introns. When we go through splicing, we cut out those introns, that pink region, and we glue the dark blue together, and here you can see our mature RNA copy that contains the code. You see how that works? All right, here's another illustration, kind of the same thing. I'll let everybody get this written down first. Introns get in the way. Exons are exciting because they have the good stuff. Or come up with your own mnemonic and how you want to remember that one. Introns in the way, exons exciting because they're the good stuff. Here's another illustration showing the same thing. We've got our DNA that's got the exons, introns. Our primary, our immature RNA copy has the introns in the way. We cut them out, glue the good stuff together, and boom. There's our mature RNA transcript, fully coded, ready to be translated into protein. Now, that sounds like a lot of work. Why not just make the genes with the exons? Don't fool with the introns. You wouldn't have to have all the enzymes. You wouldn't have to have as many nucleotides. But when we see things in the human body and in our cells, that means there must be a survival advantage. Either that or it doesn't do any harm, and so it's just sort of been kept around. But in this case, there's a survival advantage for the introns. Can you think of an advantage of having all this junk DNA interspersed between all the good stuff? Can you think of any reason why that would be good? Ooh, you need it. So it's there because you do need it. So you were close. I mean, you were close. But why do we need the junk there? Okay, let me ask you this question. My dad was a history teacher, I told you that. He loved World War II movies. His dad fought in World War II, he was in the Navy. So if it's got John Wayne in it and it's got World War II, we had to watch it. Probably 100 times. So if I'm thinking, okay, I'm in the military, and I'm told that I am going to have to go out and attack the enemy. And there are 100 enemy just waiting out there. And my commanding officer says, you, by yourself, go get them. Odds aren't very good, right? Because there are going to be 100 guns and 100 bullets coming right in one place. Now, if I've got 999 buddies with me, and we go against 100, all of a sudden, the odds of one of those guns being on me went way down, right? The bullet is going to be more likely to hit somebody else. Can you see where I'm going with my analogy here? Your DNA is under attack every day, especially here in the South and especially in the summer. What bullets are hitting our DNA? Ultraviolet radiation, sunlight. And those bullets can, in fact, lead to mutations and damage to your DNA. So do you see why we have the introns? The introns are those extra guys that can take the bullet for you. We want the mutations to happen in the introns. We need the mutations to happen in the introns because we're going to cut that junk out anyway. We want our exons to be safe. So I think of the introns as just that, the DNA that's taking the bullet, that's not going to lead to cancer. It's not going to be passed on in a sperm or an egg or in another cell that could just get worse and worse and worse. So that's why we have to go through all the headache of splicing out the introns, splicing together the exons, so we can interpret and understand the code. And that code is called the genetic code. It's composed of our nucleotide alphabet. Now, there are a lot of words in the English language. There are a lot of new words that are invented every single day. How many letters in our alphabet? 27? 26? 27? That's why I'm in biology. 20-something. In the genetic code, we have 4 letters in our alphabet. A, T, C, G. If you put it in RNA language, it's A, U, C, G. And we're going to use those 4 letters to assemble them into 3-letter words. And these 3-letter words are called codons. That is going to be the code for our amino acids. Now, when we look at having 4 letters, and we're going to assemble them into 3-letter words, that means there's 64 combinations we can have. But we've only got 20 amino acids, so that means some of these codons are going to be used for a similar amino acid as another codon. So let's look at this word, AUG. Is that in DNA or RNA? How do you know? Because there's a U, exactly. If that was the DNA gene, what would it say? A TG. So AUG is a codon that's going to be used along with the ribosome to assemble the amino acid into a protein, and the amino acid that's going to be assembled using that code is called methionine. And the abbreviation for methionine is, guess what? M. Isn't that clever? And the reason I like using that as a first example is methionine is the very first amino acid in every single protein. So, do you know what else we call AUG? We don't just call it the codon for methionine, we call it the start codon. So when you look at a gene and you see AUG, okay, that's going to be the first amino acid, methionine, but you're like, okay, we're starting the process of assembling our amino acids at our AUG. Now, this looks really, really busy, and I bet you're really glad right now you're not a biology major, because you have to memorize this chart if you're a biology major. Doesn't that sound painful? It's really not as bad as it looks. There's tricks to it. You know I like tricks. I like working smarter, not harder, but you're not going to have to memorize this chart. There are a few I want you to know. Methionine start, I want you to know that's AUG. But notice in our chart, the way this is assembled is you see our 4-letter alphabet, our 4 nucleotides on the left-hand side. This is going to be the first position of our codon. So here where we see A, all of the very first letters for these 4 different ones that start with an A. When we read along the top, these are the nucleotides in the second position of our codon. So let's go back down here. There's an A. We go up to this top column U, and there's U in that second spot for every single one. Now when we read over on the right side, you can see we've got these kind of squished in together. You see that? UCAG, UCAG for each one. That's a third position. And you can see UCAG, you see how you read this chart now? But the thing I want you to really take home is we can look at the amino acids in this one. This column, the second one from the left. Serine, proline, threonine, and alanine. Each one of those have 4 different codons that encode for those amino acids. And when you look at each one of those sets of 4, what's the only thing changing? Is it the first codon, the first letter? Is it the second letter? What letter's changing? The third letter. This third letter changes, but it doesn't make a difference because of something called the wobble effect. As long as for some of these codons, the first 2 are correct, it doesn't matter what the third one is. For serine, proline, leucine, isoleucine, threonine, valine. For most of these, you can have 4. There's glycine, arginine. Some of them are going to be a little bit more picky, a little bit more finicky. But that wobble effect says as long as you got the first 2, we don't care what the third one is. And again, there's our example of what we see with alanine. Does everyone understand how to read the chart first? Does everyone understand when you see the wobble effect here? That third codon's not making a whole lot of difference for these. Now here where we've got this upper left hand, where we have phenylalanine and leucine, yeah, we do have a difference. But notice UC in that third spot encodes for phenylalanine, AG codes for leucine. So there's a smaller sort of wobble effect there. But that third position is the one that's kind of loosey-goosey. And that's what that's all about with that wobble effect. All right, so transcription, we converted our DNA code into RNA. And we principally did that so it could leave the nucleus. And when we're talking about RNA, I want you to know that there are 3 different types of RNA that we're going to have to use in the process of translation, getting that genetic code and assembling it into a protein. And there's no real significant order to how to do this, but we have ribosomal RNA that we can abbreviate as lowercase r RNA. And guess what you use to make that? You put ribosomal RNA and it's a ribosome. Remember we got the 2 subunits, like clapping your hands together? Well, These ribosomes have a large and small subunit, and on the inside of our ribosomes, that's where the RNA that has the code is going to slide, and that's where our amino acids are going to be brought in one at a time and assembled into our protein chain. But where do we find ribosomes? In the nucleus or in the cytoplasm? In the cytoplasm, because that's where translation occurs. That's where building the protein occurs. Sometimes on the rough ER, because that's where ribosomes live sometimes just free-floating ribosomes. So ribosomes are the factory. They're the warehouse where proteins are going to be assembled. Just like workers and all the processes occur inside a building, this is the building for producing protein, the ribosome. Now, our second type, and you may not have heard about this one, but this one is called transfer RNA. And I don't know why that is off the edge of the screen here. But understand that transfer RNA does two things. Number one, transfer RNA, the name. What do you think transfer RNA is going to transfer? Amino acids. Transfer RNA is going to go out into the cytoplasm, and it's going to grab on to a specific amino acid that it's designed to bind to. And you can see in our illustration here, we have an amino acid binding site. It's not random. It's going to specifically bind a certain amino acid. We'll say that that's methionine. We've been playing with methionine already. And so our amino acid is bound on one end, but the opposite end, we have a part of that RNA molecule that is going to be essentially a match for what we see on our RNA. What was our methionine codon? Our start codon, do you remember that one? UAG. When we see UAG on our RNA, we know that's a start codon. Well, our amino acid here for methionine, it's got methionine bound to it. It's going to have a match for that. We're going to say that this codon is UAG that's in the gray there. This anticodon is going to be kind of the opposite. Here where we have a U, what do you think is going to be on our transfer RNA anticodon? What would match with a U in RNA terms? U is going to be an A. On our anticodon, we had UA, so what's going to be here? And our anticodon. What's the opposite of A? U. Remember, we're an RNA. And then over here we have a G, and this is going to be the same for DNA or RNA. What are we going to find on the anti? C. Do you understand anticodon? It's going to find its match where we have the genetic code, and that's going to help bring the amino acids in the correct order. So real quickly, we're going to assemble all these things together. We got the two parts of the ribosome. They're going to assemble. We've got our RNA. And the RNA that contains our code is called the messenger or mRNA. That's going to be the code that we're going to read. And our transfer RNAs are going to come in. So we're going to have our first transfer RNA that has this AUG. I think I got my numbers backwards there before on our illustration. But when we have this methionine transfer RNA come in, it's going to bind with our ribosomal subunit, and it's going to bind to our mRNA at that right code. That's going to be the initiation of translation. Let's look at a picture first before we do all this writing. Can we do that first? Okay, let's look right here. All right. So this thing that looks like a little coat hanger, that's going to be our transfer RNA. You see how it's got the M stuck on the top? That's our amino acid binding site. And here we have our start codon, AUG. There's our anticodon. The U matches the A matches the U, and the C matches the G. That's what assembles that in the right place. Then we have our subunits of our ribosome. They clap around all that, and that starts this process. And the ribosome is going to start sliding down that mRNA molecule. As it slides down, there's going to be a second transfer RNA with the second amino acid that's going to be able to come in and bind to its specific sequence that's next in line. And that next in line is called elongation. So our methionine transfer RNA that was sitting in its place, which is kind of in the middle of the ribosome, it's in a place that's called the P pocket or the P site. We'll look at those letters too a little bit closer in a second. Well, the next transfer RNA comes in and it sits down in the very first position that's called the A position. And I've got these mixed up. P is not the first, it's the second. A is the first. Because A means arriving. And as that new transfer RNA arrives and it sits in that right-hand-most spot with our methionine transfer RNA in the second spot, and that P site is called the peptide site. When that happens, those two amino acids are close enough together in the ribosome that a peptide bond is going to form. And that methionine is going to detach from its transfer RNA, and it's going to be stuck to that newly arrived one like the growing beads of a pearl necklace. And as soon as that peptide bond forms, then the ribosome is going to slide 3 nucleotides. And that's going to move our 2 transfer RNAs 1 position to the left. All right, so let's look at that picture. So you see what we've done here. This is the arrival site. This middle site is the P pocket or the peptide site. And then we've got a third pocket over here. It's called the E site, E for exit. So as we bring in the next transfer RNA using its anticodon, it binds. With the existing amino acids that are in the P site in the middle, that peptide bond is formed. And you see how all of the amino acids get linked together? And they're all going to be joined to that amino acid that just got there in the A site. This transfer RNA is going to let go of its amino acid, and as soon as it lets go, guess what? That empty transfer RNA gets spit out. And that spitting out causes that ribosome to move 3 nucleotides over. It reads the next codon, and the next codon, and the next codon. Until finally, we get to a codon that doesn't have an amino acid bearing codon with it. And when you reach this codon that is a stop codon, which we call the termination, then the ribosome, it's like it explodes. The ribosome falls apart, the protein floats away, and the mRNA is there so that you can do the process all over again.